Hello everyone, this is Tushar Biswas and welcome to the Study Companion. Today, as promised earlier, today we are having our third video on DNA double helix and we will be learning about the various structures, the nitrogenous basis structures and how do these structures look when they are in their nucleoside form. We know that nucleoside means sugar base connected with the nitrogenous base. The sugar connected with the nitrogenous base is forming the nucleoside. If you don't know this yet, please take a look at my first video, which will appear in the I button right now. It's going to appear in the I button, and you can always check that out. So let's begin without any further ado. Okay. So today we are going to see the structures of nitrogenous bases. Structures of nitrogenous bases. Structures of nitrogenous bases. Now let us check out the structures. First of all, we'll see adenine, the purines and the pyrimidines. If you remember, okay, if you remember, we have purines and we have pyrimidines or pyrimidines, whatever you like to say. Okay, so the purines bind with pyrimidines, and that's how base pair occurs. Now we know that, but we don't know about the structure yet. Let us take a look at the structure. Purines consist of adenine guanine. Purines or purines consist of adenine and guanine. Now let us focus on adenine first. How does adenine looks? Then we'll move on to guanine. Okay, time for adenine. Let's see. Benzene ring over here. H here. One double bond over here. One, two, three, and four. One double bond over here. One H here. Now erasing two parts of this purine. Not purine, the benzene ring. We'll put N. Okay. Now let's draw a bigger diagram. This one is a smaller one. Let's draw a bigger one. Let's draw a bigger one. Double bond over here. Over here, normal benzene rings. Double bond. Erasing parts of benzene ring. These two parts where we insert nitrogen. Okay. NH2 over here. NH over here. And it's done. This is what adenine is. Okay, guys, this is adenine. Okay, guys, so now we are gonna draw guanine. N over here, N over here, benzene ring here, double bond, hydrogen, hydrogen. Nitrogen has three hands, so one, two, three, no hydrogen over here. One, two, three, no hydrogen required. but here we require one hydrogen because 1, 2, and 3. Over here, benzene ring will have two double bonds because one of the double bond will be shifted over here due to oxygen binding. Okay, there will be N over here and N over here, N and N. Now this N, there is already two hands busy over here. The third hand will bind to one each. And this carbon over here will bind to a nitrogen, NH2 ammonia, not ammonia actually, NH2, and this 1, 2, 3. Yeah, it's done. This one has to look symmetrical a bit. Just still okay. So these were the structure of the two purines we had. Copy it down, please. Okay, erasing it out now. Now we are going to focus on pyrimidines. There are three kinds of pyrimidines: uracil, cytosine, and thymine. So there will be three boxes drawn over here for them. The purine, always remember, the purines have two structures, whereas the pyrimidines have only one structure, one ring. Purines have two rings, pyrimidines have only one ring, single ring. Okay, so first of all, we are going to draw cytosine, then we are going to draw uracil, and then comes thymine. Okay, why uracil first? You will get to know it once I draw. I have something to explain over there. Okay, so guys, time for cytosine now. Let us see. Cytosine will be in the form of cyclobenzene. If you remember the aromatic ring, the cyclobenzene ring, cyclobenzene ring. These are the double bonds. This bond is going to shift over here. This bond is going to shift over here. Let's see how it performs over here. It'll shift over here and bind with an oxygen. We'll shift over there and bind with an oxygen. Over here, as usual, we'll have one nitrogen. Similarly, over here also we'll have another nitrogen. This part and this part will be bonded to hydrogens, which in purines had been bonded to another cyclic structure. Over here we'll have normally bonded NH2. 1, 2, 3, 3 arms are taken. One arm is still left. 1, 2, 3, H over here. Yeah, that's the final structure. Okay, so time for uracil now. Again, cyclobenzene. Okay, cyclobenzene bonded, this part bonded with H, this one with H. Over here we have, first of all, this bond is gonna shift over here, forming double bonded O, so no bond over here. Again, this bond is gonna shift over here, forming another doubly bonded O. This one is gonna be a bit fat. Okay, sorry for that. Let's become a bit fat. Yep. N over here, same N over here. Okay. Now there is only one, two arms, so one each over here. One, two arms, so one each over here. And that's all for uracil. Now, guys, we know thymine is an evolved form of uracil. Okay, let us prove it down. Let us prove it down. Cyclobenzene. It will look exactly like uracil, means it will have doubly bonded over here, doubly bonded over here. Okay, okay. One double bond will be situated here. Nitrogen will be present over here with one hydrogen. Another nitrogen would be present over here with one hydrogen. Okay, and now this will be H. The place where the difference occurs will be a place where the difference occurs would be over here. It will feature a CH3. It will feature a CH3, a methane, a methylated area, not methane as such, it's a methylated area. So methylation of uracil forms thymine. Methylation of uracil forms thymine. Methylation in DNA is something where you can mute a part of a DNA. Mute, not part of a DNA, mute a nucleotide. Okay, you can mute a nucleotide, mute a base pair through methylation. This part is rendered unreactable. This won't react anymore. This is muted. Over here, this could have reacted, but here this is muted. Hence, more stability. See over here, this and this part, both of these parts could react. Any, see, all of these parts can react. Okay, this part, this part, this part, any of these parts can react. So there are one, two, three, four, five, six reacting areas. Over here there are only five. One reacting area has gone down because of methylation. This is the methyl. CH3 is the methyl group. CH3 is methyl group. Methyl group. Okay, this methylation reduces its stability, reduces its reactability, increases its stability. Okay, guys, please copy that one down. This making this diagram a bit more uh, legible, bit more symmetrical. Okay, copy that one down. And this is all about the structures. Okay, So guys, now let us focus on the most awaited part. How does a nucleoside and a nucleotide with its nitrogenous base looks in real life? Okay, let us see. First, we are focusing on adenine, adenosine actually, adenosine. Okay, adenosine. You're gonna have this sugar, 5 prime, not 5 prime, sorry, pentose sugar, deoxyribose pentose sugar, if you remember. Now branches you see, these branches you see, these branches are actually carbon branches. These branches are not interlinked to each other. There is one carbon in between. What happens is, see, this is the scenario. This carbon is linked to one hydrogen up, this one OH at the bottom. Over here, same thing here too, and same over here. This carbon has one OH at one end and two H at other ends. H can be anywhere, OH can be anywhere, provided whatever suits you. So this is the actual structure representing this. I'm representing the carbons over here. So over here, if you see one straight line, this is because there is one carbon in between. Okay, I hope everyone has understood this part. Now, moving on to the actual part. Moving on to the actual part. Let me draw a better diagram. This is my sugar first. This is my sugar base, deoxyribose pentose sugar. From this carbon, there is one hydrogen, there is one OH group, hydroxyl group. From this carbon, this is 1 prime, this is 4 prime. There is one 5 prime carbon, which is again linked with 2H and one OH group. And this is 1H of the 4 prime. Okay, now this 1 prime carbon. Now there is, for example, adenine. See adenosine, so we are focusing on adenine right now. We'll be drawing adenine right now. N, N, there is a double bond over here. Removal of this part where we'll add N, again N over here, NH2, double bond here, CH, 1, 2, 3, 4 for the carbon. And N has 3 arms taken up, 1, 2, and 3, N, 1, 2, and 3. Taken up, nothing more in written. Yeah, of course, there is one more double bond over here. This is now bonded to an H, and this N has only 2 arms taken, so the third arm will have an H. Now this is adenine, and this is your sugar phosphate, sugar base only. Right now I'm not focusing on phosphate. This OH actually, this OH will react with this H and in turn will form water and will be expelled out. Water will be expelled out. Okay, after the expelling out of water, automatically when these two things are reacting to each other and expelling out, there will be a bond formation over here. This arm and this arm will become singular again. These two arms are going to be singular back again, and the things on these two arms, hydrogen and OH group, hydroxyl group, these two are reacting, so these arms are coming together. When these two are reacting and going away, these arms are pretty close, so they'll attach with each other. Take it in that form. So that is a feel actually. That is actually the feel. Okay, that is something which doesn't happen. That happens is that this hydrogen is reacting with OH minus plus H plus will give me H2O. This my negative charge will pull in hydrogen. Hence hydrogen will, prior to this, it was in the formation of NH. Nitrogen is negative, hydrogen is positive. Now when this H is given out, nitrogen again becomes negative and hydrogen is given out in the form of positive. This positive hydrogen is this, which reacts with OH. So nitrogen is again rendered negative. Now this nitrogen is negative now. It will search for something to become stable again. Over here, this carbon has lost its negative part, its negative group. So now this carbon is slightly positive. Over here, the carbon was bound to OH. This OH was negative. Now when it's given out, carbon is now slightly negative, not negative, sorry, positive now, because all carbon has is hydrogen. 1, 2, and 3. There are three carbon has four arms. Carbon has four arms. Carbon already has three hydrogen. Carbon has, hence carbon is having a plus charge, has having a positive charge right now. Okay, it was balanced now. Prior to this, H2 I'm writing because I won't get space over here. H. It was balanced. It was balanced. It was stable. Now when the negative charges are given out, when the negative charge is given out, all the number of positive charge on this carbon will be more. There is no negative charge over here. So the positive charge will take over carbon, and hence carbon is positive now, whereas nitrogen is negative. Now this positive carbon with one free end will bind to this negative nitrogen with another free end. I hope I am making myself clear. This carbon is now positive in nature because carbon was stable. Over here carbon was stable. OH was actually minus, is OH was the negative group. Over here H is positive, H is positive, H2 is positive, this is positive, this is the negative group, making the carbon positive. Now when this OH is given out, the negative group is taken away. When the negative group is taken away, the positive group only prevails. This time, this time this positive group will have an effect on carbon, and carbon will turn positive in nature because all it has are positive groups. Carbon itself is negative, of course it's negative, but these positive groups overall will make the whole compound positive. It's not that carbon is positive, this whole compound becomes positive. Okay, this whole compound becomes positive in nature. This whole compounds become positive in nature. This is positive now, means this part has become positive and this part has become negative with free ends. Okay, these have been taken up, these have been taken out in the form of water, and now this has become, for people who had any doubt, I just erased the two and made it away. For people who had doubts, okay, now this is slightly positive because there is no OH over here. This is slightly negative because there is no H over here to make it stable. Prior to this, H was there, it was stable, it was neutral. When NH was present, NH was neutral. Okay, NH was neutral. I'm just focusing on this part. That's the reason I wrote NH. NH was neutral. Now H has been taken away, the positive charge has been taken away. Now N has slightly negative charge. N has a negative charge, whereas carbon was 1 prime carbon was stable. Now the negative charge has been taken away, so it's now positive in nature. So the positive charge now attracts the negative charge, and in turn they bond. They bond with each other, and in turn they bond with each other. I was checking my camera in case it wasn't recorded. They bond with each other. That's how. That's how. And then this positive charge and the negative charges vanish because now they are and they have attained the stability again. This positive, this negative will now react together, and the stability will be restored back again. This is how adenosine is formed. I hope everything is clear over here. If you want, you can copy that one down. We'll now move on to guanosine. Of course, I'll show you the phosphate formation, like attachment of the phosphate bundles. Okay, copied it down, I guess. Erasing this part. Now, phosphate group looks like this. There's no H actually, that's actually negative. This is negative, this is negative, this is negative. These three sites are the reacting sites. This is inert. This is inert. These three are the reacting sites. This one takes an H from the system and becomes OH, making it unreactive. Right now in phosphate, OH right now will be unreactive. In this, OH was reactive. For this one, OH is reactive and non-reactive. The reactive sites are these two now. This one binds to 3 prime. If this one binds to 3 prime, this will bind to 5 prime. If this one binds to 5 prime, this one will bind to 3 prime carbons of the sugar. These are the ones which catch hold of the sugar. One catches 3 prime, one catches 5 prime, one catches 3 prime, one catches 5 prime, 3 prime, 5 prime, 5 prime, 3 prime. Catching changes, okay. So it depends on these two things where will they attach. Okay, one OH is taken away now, one OH is removed. Okay, one OH is taken away now. Okay, one OH is taken away now, and one of the O will fit in over here. This is the P with this O up over here. This O will be over here, and this OH would be over here, and that's all. That's a nucleotide now. Now this O will again bulge into the other nucleotide up over there, other sugar, other nucleoside up over there. This is gonna bulge into the 3 prime OH and will remove the OH, place its oxygen and bind. Okay, that's how the oxygen is removed and it's bound. Okay, okay, I hope the camera is clear. Yeah, camera is clear. This is the formation of adenosine. If you want, you can copy that one down. I'm erasing this one now. Now let us focus on guanosine. Then we'll go into cytidine and everything. Now what about the nomenclature? Why cytidine? Why not cytosine? We are coming to it. Time for guanosine. The only changement over here would be removal of this bond. Okay, there is no more change over here. It's gonna be doubly bonded to an oxygen molecule, not molecule, oxygen atom, double bonded to oxygen atom, and this double bond has been removed. So nitrogen has only 2 hands right now, and nitrogen bind to one each. Over here, nitrogen has 3 hands. It's okay. This carbon binds to NH2 back again. Okay, nitrogen one hand and two other hands for H2. This is guanosine. Copy that down, please. One more thing, these are negatively charged. Huh? Change that for adenosine too. Guanosine. Okay. Now moving on to cytidine. Okay, why cytidine? Because the parents are the purines or purine, whatever you like to say, and pyrimidine. Now the purine or the purine, the purine consists of adenine and guanine. Hence, when they are in this type of formation, they will take up the suffix ine from purine and will become adenosine and guanosine. Okay, and then what about the pyrimidines? They will take the last four suffix and become cytidine, thymidine, and uridine. Okay, now we are drawing the other part, the pyrimidine, sorry, pyrimidine part. Let's focus there. Same thing will happen for pyrimidine. Same thing will happen, but the structure would be a bit different in the sense that there will be only one cyclic structure. Okay, drawing cytidine over here. This is my cytidine. There will be N over here. There will be N over here. Sure. This is my cytidine. N over here. Let's draw a better diagram. This one is looking a bit dirty. Okay. There's gonna be an and there's going to be N over here, N over here. Okay. Doubly bonded O and one NH2, one double bond over here, one double bond over here. This is my cytidine. Two arms for N, so there will be one H over here. Three arms for N, so no H required. Now this H and this OH would again form a bond, would again elope, will escape in the form of water, and the same reaction would occur. And and nitrogen and carbon in turn would get bonded to each other. Nitrogen and carbon are now getting bonded to each other. This is what is happening over there. This is my cytidine. I'm purposely drawing this as a straight structure. This would have been curved over here. When I'm drawing the actual one, this will be curved over here. This whole thing will be sleeping over. This whole thing will sleep down because there has to be a straight bond. So it will just sleep down. Okay. I'm just drawing it like this for the explanation's sake. Next, we are coming to uridine. Okay, copy this one down. I guess everyone has copied this one down. Uridine over here. There's going to be some bits and pieces of changements to a doubly bonded O and NH. This is my urea. Not urea, sorry. Not urea, sorry. This is uridine. Previously was cytidine, now this is uridine. Previously that was cytidine. Cytidine completed, uridine completed. This is uridine, mind you, this is uridine. And now, lastly, we have thymidine. Lastly, we have thymidine. Only place of change is over here. Methylation, if you remember, this was CH2 and one, not two, sorry, three. So that was CH3, and that is thymidine. Finished. This is my thymidine. CH3, huh? This is CH3. CH3. This is my thymidine. Okay, now let us draw the connecting structures. Okay, let us draw the connecting structures. This is thymidine. I'll turn it. I'll turn this one. I'll turn this one. And let's see what will happen. So now the most awaited part we have brought in over here is how the bases are reacting with each other and forming the bonds amongst themselves to give you that actual feel of DNA. Okay, the actual feel of DNA. We are going to draw adenine on this side. Yeah, this is our adenine. Let me see if the camera is recording. Yeah, camera is recording pretty good. The diagram didn't suit me. I don't like this kind of diagrams. Sometimes it happens. Yeah, now it's good. Now it's good. This carbon is bound to an NH2. There is an N over here with 1, 2, 3 arms taken. 3 arms are taken. There is an N over here too with 3 arms taken. Okay, with 3 arms taken. That's all for adenine, adenosine. Now let us draw thymine. If you remember, this side will be 3 prime to 5 prime. So the sugar phosphate will be below. Sugar and the phosphate would be below, and then there would be connection, and the thymine would be upside down. Mirroredly, it will be inverted. Uh, it will have a lateral inversion as well as it will be upside down, just because the sugar phosphate base is upside down now. Compared to this, now this has reversed. If you remember the first video, how the base pairing occurs. Over here we'll have the normal base pairings. This nitrogen bound to the sugar. Okay, I'm drawing thymidine now. There's one CH3 over here, one open end over here. Okay, one open end over here. It's doubly bonded with O. Over here, it's doubly bonded with O. And here is one N out here. This is all. This is all now. Not only an N. See 1, 2, and 3 H over here. Now this is all. This is all. This is adenine. This is thymine. Now how do they bond? This hydrogen bonds to oxygen, and this nitrogen bonds to hydrogen. That's how the bonding is occurring. Okay. Let me remove the dotted lines. Let me draw the Let me draw one single straight line. This and this. Hence, two bondings in between adenine and thymine. Two bonds between adenine and thymine. I hope this is clear. I hope this is clear. This is what is happening. This is adenine. I didn't draw the sugar and phosphate over here because I didn't have space. Of course, I didn't have space. and right now this is the 3' end of the sugar, this is the 5' end, so this is inverted. This thing has been inverted, as well as lateral deviation has taken, not lateral deviation, it has been inverted laterally too. It has become, from here it has become this, as well it has been inverted, so it's this. Okay, if this is the part for this one, first thing happened was it had become like this. Second thing that happened was that it had turned upside down. Okay, so this is what happens in the formation for adenine guanine. AT bond, AT bond, adenine thymine bond. Please copy that down. Now this can be converted into uracil. This can be converted into uracil by just the removal of this CH3. Instead, you will have one H over here, and boom. And this will now feature AU bond in RNA, specific to RNA only. AU bond specific to RNA only. Prior to this was the AT bond specific to DNA only. This is specific to RNA only. I hope everything is clear right now. Okay, now I'm drawing the next part, cytosine guanine, and with that I'll end this lecture. Okay, it's gonna be a pretty long video, so bear with me a bit. Of course, this is informative, pretty informative. Okay, in school they won't show you like this. They won't show you like this in school, actually, because in school we are limited by time, right? 40 minutes class, 45 minutes class, hardly 50 minutes can be extended, like hardly it can be extended to 50 minutes. So it's actually not possible, with the vastness of the syllabus, it's actually not possible to show you these kinds of diagrams, which I can, of course, because I'm not time bound. My focus is to give you information. Now it's up to you what you do with it. Okay, so copy this one down, copy this one down, guys, copy this one down. Hmm, and I'm erasing it. Now time for guanine and cytosine. I'll just change a part of this diagram only. Okay, I won't be redrawing it because it would be unnecessary waste of time. Okay, and you have to again rewatch stuffs like that. So I'll just be erasing the parts we are not requiring it. I'll just keep in the common parts. I'll just do the necessary changements required over there, and we are good to go again. This one looks pretty discouraging. Looks good, I guess. Yeah, kinda great. This one looks better. Yeah, I've been checking my camera right now. I have been checking my camera because I have to see now if it's focusing or not. There are times I just lose out on various videos just because my camera didn't focus, or my or my system's RAM didn't support it. Okay, I have had those bad experiences. Okay, let's now focus on the structure of guanine. This double bond vanishes. This double bond vanishes, and over here an oxygen atom is left out. Since there are only two arms, there is one hydrogen out here, and one, two. this will be doubly bonded. There will be an NH2 over here, guanine or guanine, whatever you like to say. Okay. Now changing this to cytidine, because of course cytosine is the only thing that binds with guanine. Cytosine is the only thing that binds with guanine, and this part will be erased off because there is no double bond. There are no two O's in cytidine. Cytidine. Okay. There's only one doubly bonded O with an N over here, which is again bound to. Okay. Now over here there is one NH3, NH2, not 3, sorry, NH2. Since there are one and two arms of nitrogen taken, there will be one more arm, H. We don't have doubly bonded O over here, so we'll have NH2, NH2, and there will be double bonding over here and double bonding over here, just like the cyclical structure. Okay, add this double bonding in the case of uracil and thymine also, because I had missed out actually on this structure. I'm very sorry I had missed out on this structure. And add this one in thymine and uh, uracil. I will mark that in the video too, but I guess it's better to say it on over here also. For thymine, it would have been like this. For thymine, it would have been N. Sorry for erasing this. CH3 double bond here, doubly bonded O, doubly bonded O, and N over here with H protruding, and N bonded to sugar. Okay, add this double bond for thymine. Add this double bond for thymine. Add this double bond for thymine. As well as for uracil, you know where to change for uracil. So please add that for uracil too. Okay, I had missed out on this double bond. Okay, so this is all we got right now. Let us check out the bonds. Let us check out the bonds. There are three bonds. I had gone to take my pen. Blue one. Guanine cytosine has three bonds. How three bonds? Hydrogen bonds. This H binds with this O, number one. Number two, this H binds with the N. This H binds with the O back again. See, three bonds we have now. Now we have three bonds. It's recording. Yeah, it's recording. And here we are done. Boom, finish. So I hope everyone did enjoy this lecture. This was informative. This was the most detailed video I can ever give on this topic, DNA structure, double helix, and everything. In case you want any lecture on aDNA and stuff like various types of DNA, aDNA, zDNA, and many more. Okay, so feel free to let me know in the comment section, and I will provide you with that. Until and unless commented, I won't make a dedicated video on it. I will just move on with the other experiments, Griffith's experiment, like Martha Chase experiment. Okay, Hershey Chase experiment, not Martha Chase. Martha Chase was the name of one of the scientists. Hershey Chase experiments. Those experiments will come in a shorter video where only one experiment will be explained in one single video. Okay, till then, keep studying. This is the Study Companion. This is Tushar Biswas signing off. Thank you.